name is Marin Matic. I'm a fourth-year PhD student in Scuola Normale Superiore in the group of Francesco Raimondi, and today I'm going to talk to you about the research we do in understanding the GPCR Gprotein selectivity by dissecting the sequence and structural determinants of it via structural bioinformatics and machine learning. The Gprotein coupled receptors, or GPCRs, are a group of membrane receptors with over 800 members that contain 7 transmembrane helix domains, very specific to them, binding to heterotrimeric G proteins consisting of G alpha subunit and G beta gamma subunit, which possible 4 alpha subunits, GS, GIO, GQ11, and G1213. The activation of the receptor by ligand starts a reaction that separates the G alpha subunit from the G beta gamma subunit and signals downstream pathways. The GPCRs are very promiscuous receptors and can bind many different ligands and couple through multiple families of G proteins. It is important to know that around 30% of all human GPCR coupling to G protein is unknown, as well as ligand binding to those. Their importance stems from their key responsibility for enabling multicellularity evolution by creating the intramolecular networks where it is possible by rewiring differently the GPCR Gprotein interface residue enable different coupling of GPCR and Gprotein, which in turn enables different pathways to be activated intracellularly. These pathways are activated by GS activates the adenylate cyclase, GIO inhibits the adenylate cyclase, G1213 activates pathways to ROGF, and GQ11 activates pathways to ROGF, RAC-CDC42, and PLC. GPCR have a crucial role in this signaling, and there are many unknowns about their selectivity and specificity, so they are a great interest to research. In this presentation, I will be focusing on the two key elements that we are researching our group. Firstly, I will talk about previously published machine learning predictor of GPCR Gprotein coupling called PRECOG-X, via which one can explore signaling mechanisms of GPCR. Second, I will extensively talk about the intermolecular networks that the GPCR Gprotein create to enable selectivity, which we analyzed through the first ever GPCR-wide structural analysis. So, previous efforts to develop a machine learning predictor of GPCR Gprotein coupling have been somewhat successful. We created a precog that was developed and used only sequencing structural features of class A receptors and could only predict for them. The results were mixed, as for some like Gprotein coupling, there could still be improvement in predicting. Therefore, we developed a new machine learning predictor, PRECOG-X, to tackle this issue by using the state-of-the-art deep protein representations of the GPCR. The said deep protein representations are based on protein language models called ESM, which are based on self-attention, developed on the basis of a natural language model. In the natural language model, we embed the sentence, and these embeddings then recapitulate all the long relationships between the words in a sentence, so we can understand what is the semantics of a word. In the case of a protein language model, we create a protein sequence embedding, which is a numeric representation of the sequence, and in the case of the protein, it recapitulates the relationship of each amino acid position with each other, which enables description of structure and function. Our predictor PRECOG-X is able to predict for all classes of GPCR and predict for 14 G protein and 3 beta arrestins. Comparing to previous iteration, our supervised binary classifier now predicts better than PRECOG across all of the G protein groups except GNAI3. We also developed a web server where one, by inputting the sequence of the GPCR as an output, gets different metrics like coupling probabilities, or you can visualize different coupling contacts based on the structure on the sequence, and project through the PCA of the input sequence on a reference embedded GPCR space and classified based on different either binding assays or GPCR classes to understand your similarity of the input sequence to the rest of the GPCR. To show the real power of PRECOG-X, not only to predict coupling of GPCR to Gprotein, but also be able to gauge into the mutational effects on the GPCR and the coupling that have the coupling effect because of it. We did a case study of using precogex to predict mutations while using 2,470 missense variants from ClinVar for 214 unique GPCRs. Here I'm showing you one of the examples that pop out very clearly, which is MC1R receptor D29494H mutation, where this mutation in experimentally is known that it causes the loss of the capability to stimulate CAMP levels, meaning there is an inhibition of the adenylate cyclase. In our case, this mutation causes now the switch to coupling of to GNA1 and enabling the GNA1 coupling, which goes along with the loss of the capability to stimulate CAMP levels. And indeed, these sequences in the GPCR embedded space have separated that the one that is the wild type is closer to now coupling of coupled GPCR to GNA1 and so on. To zoom in on the finer points of Gprotein GPCR coupling and to understand determinants of the selectivity of different proteins and GPCR, we have done the first ever GPCR-wide structure analysis. The understanding of determinants of coupling is a key to understanding the GPCR biology and to understand its signaling mechanism, as well as to enable us new design possibility. In recent years, a lot of structures have been resolved of GPCR Gprotein complexes with the state-of-the-art methods such as cryomicroscopy or X-ray. In each of these studies, researchers tried to point out to the specific determinants of coupling of Gprotein and GPCR, mostly for GS versus GIO. However, an overall picture of coupling determinants for all of the Gprotein family is still missing. So, to understand the determinants of the coupling selectivity, we did the first ever GPCR-wide structural analysis. We approached this problem from 2 points. First, we used the GPCR Gprotein complex structures experimentally resolved from PDB, mostly containing GIO and GS structures, with a total of 362 structures, out of which 125 unique GPCR Gprotein structures. We also did the first ever extensive AlphaFold multimer prediction of binary interaction based on the experimental datasets of GPCR with heterotrimeric Gprotein, which will be talked by my colleague Pasquale Miglionico in his poster. We approached our analysis in 2 ways. First, we analyzed the contact interfaces of different Gprotein families to understand what are the key determinants and the key interface fingerprints that separate the different Gprotein families. On the other hand, to better understand conformational differences between those complexes, we have done structural alignments of GPCR Gprotein complex by superimposing the receptor C alpha atom and calculating the RMSD of the G alpha subunit C alpha atoms. We also calculated the binding energies of the interface for each of the complexes and to understand the important and difference in their interfaces. For contact analysis of the interface, we focused on the difference between GS and GIO complexes of the experimental structures. We aggregate the interface contacts that are less than 8 angstrom apart on the secondary structure element. Green is the GPCR secondary structure element, blue is the Gprotein structure elements, and we created the networks of their contacts which show distinct difference between themselves. I also pointed out by different metrics of the networks, such as centrality betweenness, where this difference is particularly shown on in the GS on TM, ICL2 and H8 that prevail more in GS complexes compared to TM3, TM6 and ICL1 that prevail more in the GIO complexes. When we aggregate all of the contact pairs of the interface for each of the GPCR and cluster them accordingly, we show 2 distinct clusters, which one is enriched with GS coupled receptors, and the second one, cluster 2, is enriched with GIO coupled receptors. This data-driven approach using unsupervised clustering can identify specific contacts and inform us about coupling selectivity. We also provide log odds ratio of each of the contact pair compared between the GS and the GIO complexes. And the distribution of this log odds ratio for the pairs show that the GS have more characteristic higher enriched contacts than GIO. If we now zoom to different clusters, firstly in the cluster 1 to see the important motifs in the GS cluster, we can see that there are important positions of TM5 and TM6 in coupled with helix 5 of the Gprotein, and they are more enriched in GS complexes than the GIO complexes. On the other hand, the interaction fingerprint for GIO in cluster 2 shows a very important motifs of TM3 in contact with for GIO compared to the GS, specifically contact with H5 helix, where distance of the H5 helix to these contacts is usually lower in the GIO complexes than in the GS complexes. As I previously said, we also align the GPCR Gprotein complex structures by superimposing the receptor and calculating the RMSD on the G alpha subunit. And when we look at the distributions of the RMSD for the GS versus GIO, we can see there is a significant difference between them, where the GS complexes show lower median RMSD. And when we zoom in into the root mean square fluctuations, we can see that the GS complexes fluctuate less on the Gprotein positions than the GIO complexes. And this all points out, also when we structurally align these structures, to more conformationally restricted GS complexes than GIO complexes, specifically on the HN and HN5 helixes of the Gprotein. It is interesting to say that this is only done by simple structural alignment, and this unsupervised alignment really discriminates between two different complexes, GS complexes and GIO complexes. Of course, as I said before, we also calculated the binding energies of the interfaces of GS and GIO complexes with the state-of-the-art Rosetta tools, where we can see that the distribution of the GS versus GIO binding interface energies show that the GS energies are lower than the GIO, which points out to more stability to GS, and that the GS has a higher delta Sasa distribution than GIO, which points to a bulkier chains. The also interesting fact is that the structures that have resolved the same GPCRs for different Gproteins, both GS and GIO, always show the same thing, where GS has a lower energy and it's more stable than the GIO complex. This all talks about possible explanation where evolution of more energetically stable and constrained GS complex to strictly control adenylate cyclide activation is opposed to a less stable and constrained GIO complex, because likely linked to the evolutionary success of this coupling to provide redundancy over adenylate cyclide inhibition. And so to conclude, the protein language models have improved the prediction of the transducer and binding selectivity, and now we are able to predict and try to explain different mutational and variance effects for all GPCR classes and G proteins. It is our structural analysis comprehensively shows and captures common and group-specific structural features, and shows that there are different docking modes of GS versus GIO complexes, where GS having a higher enrichment of characteristic contacts and it is more conserved. These structural hallmarks lead to different binding energy of their interfaces, where discriminating between GS versus GIO coupling, and also on a class-dependent difference, where for GS A versus A class versus B1 class. So I would like to thank my colleagues in bioinformatics group, specifically Francesco Raimondi and Pasquale Miglionico, and the entire biology department, and our collaborators, and thank you for your attention.